In Chapter 18, now we're going to focus on the blood vessels that make up the cardiovascular system. So we'll look at arteries, capillaries, and veins. So as an introduction, we've already talked about that the blood vessels are needed to transport the blood to the tissues, and then of course transport it back to the heart. But we know that at the tissues, that's where you're going to have the gases, carbon dioxide and oxygen exchanged, nutrients and waste products exchanged back and forth between the tissues and the blood. Another thing though that blood vessels do is regulate how much blood is actually going into the tissues. So the rate, also known as the perfusion, the quantity of blood that's going into those tissues, into those organs. Also, we'll see with some of the vessels, they are able to control blood pressure by either vasodilation or vasoconstriction. And then finally, we know that the blood vessels themselves can secrete a variety of chemicals that are needed for the circulatory system to function like it should. So again, if we analyze both the pulmonary and the systemic circuits, we know that there are three different types of blood vessels. Each one has its own histology and its own function in the body. So we'll look at arteries, capillaries, and veins. So if you notice with arteries though, this is considered to be the distribution system. We know that arteries that begins with an A carries blood away from the heart. So as they're moving out into the tissues, they're branching into smaller and smaller vessels with smaller diameter than the previous vessels, and it's them that will supply most of the tissues found all throughout the body with the blood supply, so that those vessels and the oxygen, the nutrient, can all do the job that they're supposed to do. Capillaries are known as portions of the exchange system if you're talking about vessels and vasculature. Some textbooks describe the capillaries as the business end of the circulatory system. So we know that you have the tiny little vessels that are interdispersed all throughout the tissues. They branch out into their own networks, and the capillary vessels and the networks are known as capillary beds. So we know that at the capillary and in the bed itself, this is where the oxygen, carbon dioxide gases will exchange, the nutrients of the waste products, and because of their histology, this can happen quite quickly because the cells in the wall of the capillary are only one cell thick, and they're usually considered to be simple squamous epithelium. So one cell thick in the wall of the capillary, very easy for diffusion and filtration to take place. And then finally, the third group are the veins. So these are known as the collection system. Why? Because they're collecting the blood from the various tissues, the various organs, and as they drain from the capillary bed, they're returning that blood and the plasma back to the right side of the heart. So we know that it's going to be an opposite than the artery side. We know that the smallest veins, which will be venules, will branch into larger veins and then even larger veins until you get to the superior and inferior vena cava, and then we know that it of course enters into the right atrium. So progressively larger vessels as you progress from the capillary bed back up to the heart. And all of this by size of arteries versus veins, and the size and the thickness of the membrane of capillaries. All of this is going to be the foundation for what we'll talk about later on in the chapter and as we go through other systems in the body. So it's important to understand as an introduction to all of this that blood pressure, blood volume, some of the chemicals, how much filtration or how much diffusion that can take place in the body, and controlling the volume of blood that's distributed to the tissues. All of that's foundation is found in the three types of vessels. So if you notice, the human body exhibit in Atlantic Station in Atlanta, Georgia, they do have on exhibit where they've isolated all of the major arteries and vessels in the body. So if you notice, they're all color-coded, and these particular ones are the red arteries and the capillaries. Some of the veins are left there, and you can notice that those are blue, but they've plasticized these. And this is remarkable to look at, the diversity and also the the actual amount, the numerous quantity of all of the vessels and the capillary beds that's dispersed all throughout the body, so that all of those trillions and trillions of cells are nourished and their waste products are removed from the tissues. Now, if we start to analyze each one of these, both arteries and veins have three different layers or tunics. We know the central area of an artery and a vein is known as the lumen, L-U-M-E-N, the lumen, and then the layers are the tunics. So if you notice, starting from inside then to the outside layer, you have the innermost layer, which is known as the tunica intimita. The tunica is also referred to as the tunica interna in other textbooks. So again, I mentioned earlier that you have endothelium, so that thin layer of endothelium. We also have talked about the same layer, the inner lining of the heart, the endocardial layer of the heart, is continuous with the blood vessel's innermost layer. So again, these smooth surface, so that blood, it will cut down on friction. The cells, as they're flowing through these vessels, will have a minimum amount of friction and hopefully a minimum amount of turbulence as they're trying to get to those tissues. They also, a lot of times within this layer, will have internal elastic fibers. So remember the property of elasticity and distensibility. So we know that they will stretch, but then once they stretch to a certain degree under increased pressure, they also have elasticity to help with the recoilability. So they go back to their original size. So if you remove the force from an artery or a vein, of course it has the distensibility to stretch under the high pressure, but then to recoil, and the elasticity if the force is removed. The middle layer is known as the tunica media. So you notice you have two different layers of tissues that are in the tunica media, or the middle layer of the blood vessel. Smooth muscle cells predominate this region, and depending on which type of vessel we're talking about, there's a arrangement of different amounts of smooth muscle cells. We know that smooth muscle cells from Anatomy 1 are involuntary. Remember, they're not striated, and they're usually found within hollow organs. So the blood vessels, of course, are hollow organs. We also know that we have elastic fibers in this area that allows the lamina layer to become elastic. So again, smooth muscles are the responsibility of controlling the diameter of the blood vessel within the tunica media. Therefore, notice it says that the amount of blood that's able to perfuse or go into the organs themselves are controlled by these smooth muscles. We also know that smooth muscles are under sympathetic nervous control. So again, the tunica media is able to vasoconstrict, or to contract, and of course that would narrow the lumen and narrow the diameter of the vessel. Also, under sympathetic stimulation, when that decreases to the smooth muscles, they're able to relax, and that allows, of course, the diameter to increase, and we call that vasodilation. Then we said the outer layer of the three is the tunica externa. So notice that this is dense, irregular connective tissue with a bunch of collagen fibers. Remember, in dense, irregular, it's going in every plane, every direction. So this layer is going to give the support to the blood vessel. It's also going to prevent overstretching, especially with those high pressures in those large arteries like the aorta, the abdominal aorta, and some of the other arteries. We know that the blood's going through the lumen so fast that it can't nourish the rest of these layers. So you do have tiny vessels that nourish the tunica media and the tunica externa tissues, and these tiny vessels are known as the vasovassoria. So again, the oxygen nutrients in the outer layers, especially in these large, huge blood vessels, the cells are too far away from the lumen to get the oxygen and the nutrients, or to let the waste products diffuse back into the blood. So having the vasovassoria is going to allow it to maintain a healthy and a very nourished set of tissues in both the middle and the external layer. So if you notice in your textbook, you have the drawing of the three layers. We talked about that thin single layer of endothelium. You have the basal lamina, which is the supportive structure, and then the internal elastic portion of that. Then we said in the tunica media, notice you have those huge walls of smooth muscle that are there, and remember they're involuntarily controlling under your vasomotor nerves, which are part of the sympathetic nervous system, the diameter of the lumen of the vessel, and then that outer layer is for support. So remember, you have the dense irregular connective tissue with those large thick collagen fibers that give the framework of the vessel and the support, and then we said those layers have to be nourished, so the vasovassoria are the tiny vessels that feed into those two layers to nourish and get rid of the waste. As far as the differences between the walls, we are now going to analyze a typical artery and a typical vein. So we know, again, because of the pressure gradients and the pressure differences between those two vessels, that arteries are going to have a thicker tunica media than the veins do. And again, this is because they're controlling blood pressure and also the blood flow, the flow and the perfusion of the blood into specific organs. So it's up to the arteries. It's their main function and main job to control blood pressure, not let it drop, not let it get too high, and to divert blood to the different organs and tissues. Also, the internal and external elastic layer is more extensive, of course, in arteries than veins, and this is again because of the higher pressures that are found in the arteries over the veins. So again, some notable differences that occur between the two vessels that all relate back to the blood and the blood pressure that's in each vessel respectively. So in your lab book, you have a histological slide that shows the thick layer of the artery. So if you notice in the histology, to the left side of it, you have this big thick layer where you have the tunica interna, the lumen, the tunica media, and then the tunica externa, very thick and defined layers. And notice that the lumen is open, so the structure of the artery is keeping it open. So the artery is the big thick structure with the large lumen that stays open. Then also notice you have the collapsed vein over here. So you still have the layers, but notice that its lumen is collapsed on itself. And we know that without blood going into a vein, it does collapse. So you can test that on yourself by pushing at the veins on the top portion of your hand there at your lower extremities, and as soon as you let go of that vein, it pops right back up and blood begins to flow into it again. So histologically it relates back to their function. And then if you notice, I'll go ahead and point out, you have this tiny little structure where you do have a lumen, you have some of the structure around in the wall, and that would of course would be a tiny capillary. Okay, so now if we're focusing on just the arteries, we're looking at the structure of each type and also how each one functions. So the thickness of the arterial wall is going to depend on the size of the artery and its function in the body. So we're able to classify arteries based on structure and function and place these arteries into three different classes. So if you notice, the first two are on this slide. You have elastic, also known as conducting arteries. So again, if you think about elastic arteries, these are the large diameter arteries like the aorta, like the abdominal aorta, the branches off of it, which would be the common iliac artery. These again are very large and they're under very much high pressures. So these are the ones that are usually near the heart, under higher pressures, and they're needed to be supporting those higher pressures, so the elastic fibers in it are very much extensive. Then you have the second group, which are the muscular arteries. If you'll think about them as distributing arteries, so muscular distributing because they're actually the intermediate in diameter of the three. their walls are predominantly smooth muscle cells, and they're the ones that branch off of the aorta into muscular arteries, and they're named according to the organ that they supply the blood to. So of course this would be like the splenectic artery, the renal artery, and the gastric artery, the hepatic artery. These are all going into the spleen, the kidneys, the intestines, and the liver respectively. The smallest of the arteries are known as arterioles. So these contain all three layers of the blood vessel, but these layers, even though they have the tunica interna, the tunica media, and the tunica externa, they're extremely thin. So notice there's only 1 to 3 layers of smooth muscle in that middle layer, and then the arterioles will branch off close to the capillary bed and directly feed into the capillary bed using a structure known as a medarteriole. So a medarteriole brings the oxygenated blood into the capillary itself. Notice that the smooth muscle cells within the medarterioles are in circular patterns, and they create something known as a precapillary sphincter. Well, we know from Anatomy 1 that sphincters are circular muscles. They surround lumens or hollow organs, and they control the entrance and exit of substances through that. So they encircle the medarteriole and capillary junction, and we'll see later on that the blood has to be dispersed to the tissues that are very active. If all of the blood was dispersed to every cell and every capillary at one time, your blood pressure would bottom out, and of course you would go into shock and die. So the body is constantly shifting the blood into individual capillaries and making sure that tissues that are very active are well supplied and well perfused with blood. So we know that we said that the blood pressure is going to be the highest in the arteries, and it's higher closer to the heart. So the aorta would be the highest pressure. So we know that there are certain cells, certain receptors that are within these arteries that are going to detect not only blood pressure but also detecting chemicals in the blood. So if you notice, by definition, baroreceptors are the ones that are looking out and guarding against huge pressure changes. So you find these within the aorta and also the common carotid artery as it comes off of the aortic trunk that's located in the neck. So that's going to maintain and make sure that the pressures in the aorta and also in the common carotid artery that is of course feeding into the brain tissue, where we have talked about in Anatomy 1 that brain tissue 4 to 5 minutes without oxygen and nutrients, it begins to die off. So again, the pressures are regulated by baroreceptors. Now, as far as the chemistry of the blood in the carotid artery and also in the aorta, you have chemoreceptors that are dispersed there. So chemoreceptors are responsible for detecting changes with blood oxygen, also carbon dioxide in the blood, and we'll see later on that carbon dioxide and oxygen have to be at homeostatic levels. You can't have too much oxygen or too much carbon dioxide. Either one would cause problems. And also it's measuring pH, which of course is how many hydrogen ions are being kicked off of these solutes that are dissolved in the plasma. So again, it's maintaining your pH levels at 7.35 to 7.45. So slightly alkaline, but a very narrow range of homeostasis for pH. So chemoreceptors and baroreceptors are very important for keeping the circulatory system in check and in homeostasis. So again, this figure shows you see the ascending aorta and the aortic arch, and there you have the chemoreceptors in the aorta and also up in the carotid artery. So a branch off of that is actually the brachiocephalic. It branches into the right common carotid and then the right subclavian. Now if we look at veins, again based off of structure and function, we can classify them with these following characteristics. So we know that typically the veins are going to outnumber the arteries because again they're going to try to drain all the blood from the tissue and get it back to the right side of the heart so the whole cycle can start over again. On average, they're going to have larger diameter within their vessels, but again, the consistency of the tunics are going to allow it to collapse if there's not pressure from the blood flowing through a vein. So 70% of the total blood that the body is going to try to move is located in the veins at any moment and time. So this includes the systemic and the pulmonary circuits within the body. 70% of the total blood volume is going through the veins at this particular moment in time. Again, veins as a function are going to function as reservoirs, so they hold blood and they can divert from veins to other parts of the circulatory system. So notice with veins as their major characteristics and structure, we've already said they have much thinner walls and the tunic layers are much thinner. They're going to have fewer elastic fibers because again they're not under very high pressures. They're going to have less smooth muscle, so they're not going to be so much involved with vasodilation or vasoconstriction, which we call vasomotor tone. And we know that their lumens are larger than arteries, and we already said earlier that they're going to outnumber arteries. So if you look at blood distribution, your systemic veins, those that are all out in the organs and the tissues, 55% of your total volume is found there, 6% in your right portion of the heart, and 3% in the pulmonary arteries. Notice your pulmonary veins. Remember, these are your oxygenated veins. 15% of the blood is there, and then 10% is in the systemic arteries. So that leaves then the capillaries on the left side of the heart with very low percentages, and the pulmonary arteries with a low percentage at 3%. If we look at the structure and function of veins, we know veins, like arteries, can be classified by their size. So now we're starting with the capillary bed and we're going back up to the right side of the heart. So we know that venules are the smallest veins. They're going to drain the deoxygenated blood and the waste products out of the capillary beds. We know that you have very tiny, very minute postcapillary venules that has nothing but just endothelium and a little bit of connective tissue, so very, very thin structure. This is going to allow these postcapillary venules to also be able to exchange material with the tissues and the interstitial fluid. But as we move from the venules to the other veins, you're going to see more distinct tunics, and as they merge, they're getting larger and larger to their lumen until you get to the inferior and superior vena cava. Most veins have that thin tunica media. We've already said very few smooth muscle cells that are there. It's not responsible for vasomotor tone, so vasoconstriction, vasodilation. The diameter is only going to change slightly under those vasomotor sensations from the sympathetic nervous system. We know that veins have valves, these venous valves that work just like the heart does. It keeps the blood from pooling in our lower extremities especially, or in areas where there's not good circulation, and it keeps a one-way flow. So again, these are extensions of the internal layer, the intima or interna layer. They overlap, prevent blood from flowing backwards, and of course people that have weak venous valves will wind up with problems with their veins, and the term for that are varicose veins. So especially in the legs, these are the most numerous veins, of course, because gravity is pulling down and the venous blood is trying to be propelled back up to the right side of the heart. So your textbook does a pretty good job with giving you the layers of the tunics, describing the structure of the three types of arteries, the smallest of the veins, which are the venules that lead out of the capillary, and then, of course, varying degrees with various larger lumens. Again, as we travel back up to the superior and inferior vena cava, you have larger and larger veins. And then also the function of each one of those. So in this section, of course, we looked at the functions of blood vessels, and we described there's four major things that blood vessels are responsible for doing. We looked at the functions of arteries, veins, and capillaries. We know arteries take blood away from the heart. We consider it to be the delivery system. Veins are the capacitance vessels because they're holding the blood. They can store the blood, but then they're trying to get it back up to the right side of the heart within the pulmonary circuit. And then capillaries, we said this is the business end. So this is where the connection between the arterial side and the venular side of the blood flow. And I mentioned earlier that it was the business end of the circulatory system. So all of the exchange with the tissues happens in the capillaries, or those very, very tiny venules. We discussed the three tunics within a blood vessel wall, and then we looked at the structure and the function of those tunics and the function of the three types of arteries. We did the same thing for the veins, and then we defined finally the baroreceptors as being stretch receptors, so it detects changes in blood pressure. And again, in the carotid artery, the aorta, you find those. You also find chemoreceptors that are responsible for maintaining three basic things: pH, number one, then carbon dioxide, and yes, oxygen is important, but in this list it takes number 3. So for homeostasis to occur, the chemoreceptors are monitoring hydrogen ions and pH, carbon dioxide levels in the blood, and also oxygen levels.